Assalamu alaikum, dear student. Welcome back with the last part of this course, uh, Recombinant DNA Technology. So, in this chapter or in this part, we will try to explain the uh, start of the recombinant technology, then its application uh, in animal genetics, and with reference to biotechnology. Okay, in early 16th, soon after the Europeans arrived in the New World, that is the America, uh, definitely it is referred to the Christopher Columbus arrival in the America. A new disease outbreak took place, and that was of the tuberculosis. Now there was a debate in the history about the source of this disease, whether it was the European community who invaded the America or whether it was local born. In 1994, a pathologist, Arsus, and a molecular biologist, and his name was Sallo, Welmer Sallo, they jointly tried to resolve this controversy and pretty much extent they were able to do it. So basically, they detected a 97 base pair piece of the uh, tuberculosis pathogen in a woman whose body was preserved, and from her lungs they got the specimens and they were able to identify the tuberculosis pathogen. And the technology that was used in that identification was the PCR, polymerase chain reaction. So this is the biotechnology part, or the recombinant DNA technology part, which played some important role in the discovery of this or solving of this mystery. But another theory later on was also considered, and that was through the snails who traveled from Africa to the America, and those snails they carried this pathogen. This is another theory. But so far, the proven one with the help of the PCR is the uh, first one which I have described. Some of the basic concepts, Stanley Cohen, he was from Stanford University, and Herbert Boyer from the University of California School of Medicine in 1973, the first time introduced the recombinant DNA technology. And basically, they produced a chimera, which is a combination of two different phenotypes. And now, this technique is a set of molecular techniques for locating, isolating the DNA, altering the DNA, and then studying this DNA segment in terms of its functions. So this is altogether is called the recombinant DNA technology. So it has a wide impact on the society. It altered the studies of the genes and consequently a green revolution, and later on the specific genes cloning and then expression in the plants, it was studied, and it resulted in the manifold increase in the production and protection against diseases of the crops. And with the help of this technology, it is possible to study the nucleotide sequences, which are basically the building blocks of the DNA. And it can create mutation very accurately and very precisely on a given spot, and we would be able to study the results of these alteration or mutations in terms of the functions of the genes. The information about the structures and the function of genes are elaborated in this way, and this technology first time it explained and revealed that there are certain non-universal codons that exist in the mitochondrial DNA. So generally we have the universal codon for each of the amino acid, which is the building block of the proteins, and these are triplet 3 nucleotide bases. For example, ATG is for the methionine. But other than these universal codons, there were some non-universal codons identified with the help of the recombinant DNA technology. So similarly, many eukaryotics are multicellular organisms. Their genes they were interrupted by introns, that is through with the help through the non-coding or with the coding regions, and this discovery is also due to the recombinant DNA technology. And finally, we are able to produce the commercial products, and one of the widely used such product is the insulin against human diabetic condition. Now, when we're working on the molecular level, most of the most powerful microscope is not able to read a single nucleotide. Rather, we can see the threads of the probably with the help of the electron microscope, we would be able to see the tiny threads, individuals, that is chromosomes. But still, the single nucleotides they won't be able to be seen. Now, for instance, we want a certain gene of the human and want it to be expressed in the bacterial cells. Then, first instance, we need to find the desired gene, that is our target. And this is a quite complicated stuff because we, for example, if we talk about the human, there is almost 3.2 billion DNA, sorry, nucleotide sequences. And a gene, typically it is around 900 or something like that. Coding region is about that base pair. So you can see it's from the millions of the part of the genome from where you have to dig out the specific gene. And itself is a very serious and tough going business. So this is first step. Even we are able to locate that gene. The next step is how we can get it separate and how we can get it cloned and then insert into the bacteria, that is the cloning part, and then how it would be successfully replicated and translated. So this all is the procedures or the protocols which requires extensive experimentation to do that. Now, the location of the specific DNA sequences. These are using the help of the cutting DNA at precise location, and then the amplification of the specific sequences, and it could be billion of times to have the whole genome sequence. Then finally the alteration through the mutation, then the joining the fragments, and then transfer it to the cells. All these are protocols and procedures for the recombinant DNA technology. Now the first part comes the cutting of the DNA, the whole genome DNA, and for that we use the restriction enzymes, and they basically recognize the specific pattern of the DNA nucleotides, and based on that they cut sugar phosphate backbone of the DNA, and DNA is uh, cut into different chunks or different fragments. And this all recognition is based on the specific nucleotide patterns. Now you see on the right side of this slide, just now in this red box, you can see there are three different type of enzymes Then the enzyme activity is given over there. Type 1 enzyme, they have cleavage and methylation, and they require ATP. So if the DNA is having the methylation after cleavage, they are able to add a methyl group to the DNA for its protection. And all this activity require the extra energy in the form of the ATP. And they have random sites and distant from the recognition sites. So recognition site is somewhere else and the cutting site is somewhere else. The second type of they can only cut and they do not require, and they generally have the recognition site, cutting site within the recognition site, cleavage site or cutting site. And the third type is the cleavage and methylation. Again, they have their function and they require ATP. So where there is cleavage and methylation, then it requires the ATP. And again, they have the random sites, and generally they cut near to the recognition site. So there are three different classes of the restriction enzymes, or enzymes which are able to cut the DNA. Now there are more than 800 different restriction enzymes that recognize and cut DNA at more than 100 sequences isolated from bacteria. So 800 different type of enzymes are available commercially now, which are isolated from the bacteria by different companies across the globes, and they are marketing it now. Now sequences recognized by the restriction enzymes are usually 4 to 8 base pair, and generally it's 6 base pair. Here in this case, on the right side you can see such example. So most recognition sites, they are basically palindromic, means the plus and minus strand of the DNA, they are truly duplicate to each other, they are joining complement to each other, and their sequences, they are palindromic. Reading from one side from left to right is meaning same as reading from right to left to the bottom strand. That is the basically palindromic. Some of the enzymes may suggest cut cohesive ends and other produce the sticky ends. For example, the PBU2 cuts in middle of its recognition site, producing blunt end, non-sticky ends. Here in this example, in the box you can see, so there are 6 base pair recognition site GAATTC. Now palindromic mean you are reading from left to right GAATTC, and just we reverse it from right to left, again the sequence is same GAATTC. So that is basically palindromic. Now the recognition side is these 6 base pair, and the cutting side is basically between G and A. So you can see at the bottom and at the top strand it will cut between G and A, and this is going to produce a sticky end because there is an overhang of 3 nucleotide bases you can see in this figure, that is TAA or AAT on the top strand, and that is basically sticky end. Now another type that produce the blunt end, and it has recognition site C, G, and it's cut between C and G, and they produce the blunt end because both the cut part and there is no overhang like structure in this, and blunt end enzyme, example SMA2. Okay, so here a list of the enzymes, sorry, in this side, and you can see the results whether it is going to produce cohesive or the blunt end, and there are 800 or more than 800 enzymes as I previously told you. Okay, so you can see hand 3 enzyme, it is cutting the one fragment of the DNA, and it's producing the overhang of the, so recognition site is between cutting site, while the recognition site is AAGCTT for the hand 3. So it is going to produce the overhang. Contrary to that, you can see the PBU2, it produces the blunt end. Now, there are two different fragments of DNA which are cut with the help of the hand 3, and after cutting, they are joined together. The two different parts of the two different sources of the DNA, they are joined together, and here you can see in the colored form, in the blue and red, they are joined together, and then they are joined with the help of the ligase enzyme. This is how we join, cut and join the two different fragments of the DNA. And this is we are going to do with the help of the vector. A vector is a circular DNA which is present in the bacteria. It's the source, basically, is the bacteria. Now on the left side in A part you can see the linear DNA. It is cut with the help of the hand 3 and it produces the different sizes of the fragments. Same is case with the circular DNA, and that's the vector, and you cut it with the hand 3 enzymes, and again you get the different type of fragments. So that means there could be more than one recognition and cutting site. So here in this case there are 4 cutting sites in the circular DNA for the hand 3. Okay, then these fragments are run down the gel in the tube. From tube they are transferred in the gel and they run from negative to positive as the DNA have the negative charge, so they are attracted to the positive charge and eventually running on the gel, and then they are separated, and you can see under the UV light, because there is dye which is used for loading the DNA, and that dye will illuminate under the UV light, and now you can see the different fragments which are cut with the help of the enzymes, and these fragments later on could be used to join together according to your requirement, seeing the size of the band, and generally we use a marker here indicated which indicates the size of the fragment produced. So this was the part 1 of the chapter 6, our last chapter of your course. Thank you very much. Allah Hafiz.